All right, take it away.
Thank you. Thank you very much for having me here and our company today. Can you hear me? Yep. No. Right up here? Okay, I'm getting it. I'm learning on this one. Okay. So, DatChem. DatChem detects breast cancer risk years before imaging can. We have a cloud platform which reads tissue chemistry, and we use an installed base of MRI scanners. The problem is that current imaging finds tumors. It actually can't see a developing cancer. High-risk women live in a cycle of surveillance currently with no objective early warning. DatChem breast can actually see a developing cancer. We have 97% accuracy in a 12-year longitudinal trial, of which the first 10 were blinded. So how do we do it? Cancer changes the tissue chemistry before it changes the tissue structure. And we actually worked out how to do this using a cell model, a very detailed cell model of tumor development, where we were actually seeing where specific genetic changes were occurring. It then took us quite a few years to translate this to be able to collect the same information in a clinical MRI scanner. Now, the gap is up to 12 years before a tumor is visible using conventional imaging. That number surprised us, but it's actually correct. So how does this work? The solution is it's an objective test for risk and for cancer. We acquire the data. It's a defined protocol. It's our protocol on a standard 3T MRI. The data goes to our cloud platform and compares this data against an exclusive database. And then the woman becomes her own control so that we can actually see her individual trajectory. So the evidence, we've completed the 12-year longitudinal study at 4 centres. We have the centres Adelaide, Brisbane, Barcelona and Newcastle. Some of these women have had up to 8 scans. Every woman is compared to her own prior scans. The high-risk cohort was over 300 women, and we have a sensitivity of 96%. What was a little more confronting was of the controls of women at low risk. We have 27 that are in transition. They're on their way to developing a cancer. We have predicted that 8 of those women actually have a cancer, and 2 of the 8 already have a confirmed malignancy, 8 and 3 years ahead of conventional MRI. We actually correctly, of the 300 women at high risk, we correctly predicted 31 out of 32 cancers. So why is this defensible? We've got a capability that would take a decade for others to reproduce. We have our data, we have our IP, we have granted patents in the US, EU and Australia. We've got unique clinical databases collected over 20 years. And we have a formal relationship with Siemens to release in the USA and Germany. And in Australia, we have a national radiology company to release our program. So regulatory and reimbursement, it's staged. We've already had multiple meetings with the FDA. We will be working towards a 510K. The timeline for the Australian TGA is around 6 months and FDA 6 to 12. We are currently completing our quality control documentation in the cloud. That has to be done before we can submit. Now, a current reimbursement for the sort of technology that we use, which is spectroscopy, is around $405. So what we have here is a revenue projection for breasts for the US market launch post-FDA. There's 26 million who have an annual MRI in the United States, and an uptake of 1% penetration would generate $100 million per annum, just to give you an idea. Now, we also have DatChem Neuro. This is the first objective test for trauma, blast exposure, blunt trauma, chronic pain, PTSD, and ADHD. Each of these conditions is different. We can distinguish between them. And the neurochemistry is gender dependent. Women with PTSD are far more affected than men with PTSD. And just to give you a touch of the ADHD, one of our psychiatrists walked in a year ago and said, this is now rampant around the planet. We have to develop a test. We can determine which neurochemical pathways are dysfunctional, and we can also determine which pathways are fixed by lisdexamfetamine, and it's usually 3 out of the 5. So our funding history, I'm Carolyn Mountford. I'm one of the co-founders with Peter Malika, a surgeon. All our IP is consolidated into DatChem. We have had over 45 million non-dilutive grant funding, and underneath are where we got the grant funding from. That's a form of peer review. Our team, our coding and cloud experts are actually an ex-ASX 200 team, and we have a US-based bioinformatician. We have got private practice radiologists and private practice surgeons to help us drive the release of this, as well as Siemens Healthineers. So our traction to date. We finished the software as a service analysis platform. It's operational. Our PTSD BLAST trial is completed. That was actually funded by US counterterrorism. We've got our longitudinal breast study completed. We have had communication with the FDA and the TGA. We have MRI scan time secured at multiple sites in Australia and Spain. And Siemens has formalized their relationship for a USA launch. We are hoping to raise $5 million to reach 3 hard milestones: regulatory approval, and commercial, the first 10 paying sites in the USA, nationally in Australia, and we need key hires for US office marketing and cloud maintenance. We have a very strong patent portfolio. Our patent attorney is New York based, and I will finish there. Thank you, and hope to answer your questions.
Great.
Very good. Do we have any questions online?
I do have a question. This is great. I love longitudinal data to predict. You mentioned about your sensitivity, but you didn't say the specificity. What is the specificity for that data sets? That's number one. Second is just make sure I understand. So you're saying, I guess, how many scans, how many years of scan would be sufficient for you to give that prediction?
So I'm going to answer your second question first. We actually predicted on the first scan of every single woman in the study, which surprised us. There is an explanation partially for that. All women carrying the BRCA1 and BRCA2 genes have the first form of dysregulation. It's there in them all. That's published. Then there are a series of other changes that follow. As far as the specificity is concerned, we can't provide that at the moment because of those women, those 450 odd women. We've actually got 71 that we are monitoring on an annual basis. So we can't actually give you that statistic until we've waited to see their outcome. We may not get their outcome once we've got TGA and FDA approval because they may well then decide that they're going to have surgery.
Oh, right. But with that said, so 10 years, right? Also, I guess you could apply to historical data, right? So this is what, maybe I misunderstood. I'm assuming you have at least 10 years data, right?
Yes, we do. So each individual patient, some of them we have been studying for 12 years, and some of them we've only just started in the last couple of years. So it has been an intake of so many women each year. So in reality, to answer your first question from before, we're probably going to need to wait the next 10 years.
Okay. Yeah, you could, have given 10 years, how much? Because you mentioned you able to detect the first scan, which is quite interesting. I would love to learn more about that.
So the stages of dysregulation are, as I said, from what we recorded in the cell model of tumor development. So we first see the lipid and cholesterol changes. and all of the women carrying the BRCA genes have those. Then
I see.
then we will see a series of metabolic changes. Then we
I see. I got you.
then we'll actually see a tumor promoter. There's actually two of them, one's systemic and one's directly related to the tumor development. And that is associated with the P52 gene becoming recordable in the cell model. Once the woman is at that stage, she's at the point of no return, and then following that, we will actually see choline, and it's well established in the literature that when there's choline in the spectrum, there are cells doubling.
Okay. Now just clarify the cohort you used, the one you mentioned, the 400 something. Are they all coming like women with certain age, or this have some predefined risk
So
for that cohort?
100 of them, according to the IBIS scoring, were low risk, and we call that our control cohort. The others were in high risk according to the IBIS scoring system, and these were women that were attending one of 4 high-risk breast clinics.
Okay.
All right. Thank you very much to Carolyn.